Thank you everyone for joining us tonight. It's a real pleasure to be able to welcome you to San Francisco State's Romberg Tiburon Center. My name is Karina Nielsen, and I'm the director of the center. I'm also a professor of biology here, and I'm a marine ecologist by training. I wanted to just say a few words about the center. For those of you who are not that familiar perhaps with us, we are, as it turns out, the only marine and coastal research center that's actually on San Francisco Bay. We are also very fortunate to have two wonderful on-site intellectual partners here in the scientific enterprise. We have a branch of the Smithsonian Environmental Research Center that has a laboratory group here, and we also are the headquarters for the San Francisco Bay National Estuarine Research Reserve. Our institution here provides a gateway for university-level researchers and students to study, conserve, and restore the amazing diversity of marine and coastal environments we have right in our own backyard. We also host really outstanding public programs like the one you're about to see tonight. I'm sure you're going to enjoy it. In addition to these public programs, we also host a lot of extended educational opportunities in the community and try to engage people in understanding more about the marine sciences. Most recently, I'll just give you one example, because we really had a lot of fun with it. We hosted about 58 teachers from the San Rafael City Schools in a really wonderful professional development program to support their implementation of next-generation science standards. We had them all out on the sailing research vessel called the Derek M. Bayless, and we conducted investigations on the bay, looking at water properties, food webs, marine mammals, and seabirds. They had a really rich experience because we partnered with San Francisco State Center for Math and Science Education and with educators from the San Francisco Bay National Estuarine Research Reserve. The graduate students from RTC who are doing research here also came out on the boat and helped the teachers understand a little bit about their projects. And assisted them with learning about how to sample and use all the different equipment and think about questions that they might be able to work on with their students. So that was a really, really wonderful experience. I'm also really happy to be able to share with the community here some of the recent commitments that San Francisco State University has made to supporting the center. I know that we've been here in the community since 1978. And we're hoping to start really a new wave of improvements out here. And President Wong, as president of San Francisco State University, has recently allocated about $2 million this year for physical plant and safety upgrades. And we expect the bidding process for some of those projects to begin within the new year. I also want to extend and introduce to the community and make a very warm welcome to our new dean of the College of Science and Engineering, Dr. Keith Bowman. If you would maybe just say a word. Say hello. I also want to acknowledge that what I have just said about the investment is true. It can be verified and audited by the CFO of the university, who happens to be here too, Ron Cortez. Would you wave your hand? So, thank you very much. At this point, I want to just also really acknowledge the huge investment and opportunity that has been provided to us by the generosity of Richard and Barbara Rosenberg, who have supported Romberg Tiburon Center with an endowment that allows us to bring you programs like this, as well as our Discovery Day, which we invite you to participate in April. Some of you are familiar with that. Barbara and Dick, unfortunately, could not join us tonight, but Barbara usually helps us open the program, and she sent a few words, so I'm just going to read those. We are very sorry we cannot join you for what should be an extraordinary talk on a critically important subject. Dick and I have a conflict tonight with the awarding of the UCSF medal to an individual whose signature program Barbara chaired, it says I chaired, I'm reading her words, and on whose foundation board Dick serves. Please accept our best wishes for a highly successful evening, and it is obvious that the programs of this high caliber, we are going to have to find a larger venue. Barbara Rosenberg. So, thanks to Barbara and Dick for their support. It is now my immense pleasure to introduce our guest speaker for tonight, Dr. Drew Harvell. She comes to us from Cornell University, where she's a professor and curator of invertebrates in the Department of Ecology and Evolutionary Biology. Professor Harvell has had over 130 major peer-reviewed publications, including in the top-tier journals Science and Nature. And she has a new book expected out in February of 2016 to be published by UC Press, and it's called A Sea of Glass. I think she'll be telling us something about this project during part of her presentation, so I'm not going to steal her thunder, but look for the book. I'm sure you'll enjoy it. Drew Harvell is one of the foremost experts on diseases in marine organisms, a subject that is not studied as well as it should be, given the likely importance of diseases influencing the abundance of any and all of the plants and animals on our planet. Just consider the condition of human populations and lives before vaccines and antibiotics were used in medicine, or when Europeans arrived to the shores of North America and exposed Native Americans to the smallpox virus. You can get the sense of what we may or may not know about marine organisms and their relationship to diseases. In a 2002 review paper that Drew is an author on, she and her co-authors concluded, in a very prescient way, that the links between climate change and disease will increase the severity of threats associated with climate warming. Here we are in 2015 with a very warm ocean, and a very warm bay, actually, and many unusual things are happening. In addition to this epidemic afflicting sea stars, which we're going to hear some more about tonight. Species are appearing further north than usual. Extensive algal blooms of a diatom that produces domoic acid have been prevalent all summer. And most recently, in the news, you've seen health warnings about eating red rock and Dungeness crabs, and it may delay the opening of the iconic Bay Area crab season. You may or may not be able to put them on your Thanksgiving plate. If anyone you know has doubts about the uncertainties we all face or continue to face as we continue to burn fossil fuels at the rate we are now, or continue to cheat on emissions testings for cars, just have them check out the ocean this year, okay? So, the decline of sea stars in this epidemic touches anyone who has spent a low tide on the beach exploring tide pools, the incredible diversity of life in them, the microcosm of the ocean, and what it is that's attracted a lot of us who love the ocean to studying it. This evening, I am especially excited to know that we are graced with the wonderfully evocative paintings by a local artist named Ellen Litwiller. If you could just wave your hand, I know she's here. The paintings that she did were inspired by her awareness and personal observations of sea star losses we are all experiencing. If you haven't already had a chance to look, I hope you'll pause to look at them at the end of the program. I'm inspired, though, to know that we have so many different ways to communicate about the importance of marine life. And I'm so heartened to know that people from so many different walks of life share this affection and wonder and awe about marine creatures. It's a reason for optimism, even in the face of the increasingly apparent challenges facing marine and coastal ecosystems. Knowledge is the first step in the pathway to progress and change. And it is with that optimistic thought in mind, please join me in welcoming our special guest tonight, Dr. Drew Harvell.
Thanks, Karina, for that amazing introduction. You've certainly sort of opened the discussion on some of the things I want to continue to talk about. And thank you all. This is an amazing turnout. I'm really delighted to see your interest, the engagement of the community, pretty amazing engagement of the university and administrators to be here for this. And so it's really a wonderful honor for me. And then finally, Ellen's evocative, beautiful artwork is a real honor to have on the walls here. So this is a event that sort of took us all by surprise, even though in some ways we had predicted things like this were likely to happen in our changing oceans, especially under warmer conditions. Nonetheless, the scale and the magnitude of this event certainly caught the public as well as all the scientists by surprise. So I'm going to talk tonight about the work we've been doing over the last 3 years now. I don't know if you're that familiar with how long this has been going on, but it's been it's been kind of a long-term event. And before I go any further, I just want to introduce a couple of my collaborators because they've been absolutely key in our ability to get this work done. So I'm a university professor, so I'm always working with several PhD students and undergrads. This is Morgan Eisenlord here, who's a student that started working with me during this event because it happened so quickly and has been involved in almost all stages of it. Maya Groner's done a lot of the statistical work as a postdoc, and Ren Yoshioka is a incredibly talented former honors student in my lab who continues to help with the work. So tonight, this is a general organization of where I'd like to go. First, I'll talk about what is killing the sea stars, the information that we have about this outbreak, what we know so far, information about whether the environment plays a role and what we've learned about the role of the environment, and then some of my thoughts about how this affects our living biodiversity here. Probably a lot of you know already that this outbreak has had a lot of public attention. In fact, the correspondent who did these video PBS NewsHour ended up winning an Emmy for her reporting of this event in the last year. There's a brewery in Oregon that developed a wasted sea star purple pale ale in support of research and sort of widening public information about this. And you know, as a scientist, a lot of the things that we study don't get this kind of attention. And so for me personally, this has been a really kind of a tough event in that I hate seeing these iconic, really ecologically important species go down. But it's helped a lot to know the public has cared so much about it and been as involved as they have. And that's I guess, been a bright light in this. So a little bit about sea star wasting disease. I'm afraid it really is pretty much as grim as we're sort of cartooned here. Essentially, what happens is these stars begin showing signs of twisting, and then their arms fall off. So it does end up being a fairly gruesome kind of process. How many of you have seen this, you know, on your beaches or around here? Yeah, so, you know, a lot of you have experienced this. It turns out it's not just that it's, you know, an interesting, concerning issue on our shores, but it has sort of wider significance, and this is the largest marine wildlife epizootic that we've experienced in the oceans. So that's a pretty big thing. A bigger one than this is white spot syndrome, which is a virus that affects shrimp, but that's a farmed species. So this is actually, we'd call this a wildlife. And it's so large, not just because of the extent of the outbreak, all the way from Mexico to Alaska, but the fact that over 20 species of stars are affected. And it's not just the West Coast, it actually began on the East Coast. And unfortunately now it's spread beyond our shores to China and reportedly also to Australia. So that means it's now called a panzootic when it reaches other continents. At least 20 star species are affected. And then finally, these are ecologically important. So the term keystone species, which is kind of a cornerstone concept in marine ecology, was actually coined to describe the activities of a couple of the species of these stars here. And it's because they have a disproportionate impact on their community. And that's obviously an issue that we're interested in following up the absence of many of these stars. So I just want to introduce a few terms. If I start talking about them as hosts, this would be the star. This is the organism that becomes diseased. A pathogen, in this case, I'm only going to be talking about things that are infectious, so that would be a bacteria, a virus, some kind of a parasite, some kind of disease-causing agent, and then the environment, everything that interacts with the pathogen and the host. And it turns out that for a lot of our outbreaks in the ocean, there's a very important role that the environment plays. A lot of these sort of disease events just kind of sit quietly ticking along until the environment changes, and then there can be an outbreak such as we're experiencing. in this case. So disease is sort of the intersection of these three things coming together. I'll talk about prevalence as the proportion of the population afflicted, and severity is so like out of 100 individuals, 5% would be, 5 out of the 100. And severity is the degree to which an individual is afflicted. And then we'll talk a little bit about differential susceptibilities. So the kinds of signs that we see if you're on your beach and you're wondering, and by the way, this still is ongoing. So this epizootic began in 2013, and it continues to this day. I've been getting reports also from the east coast of stars still dying there, and the stars on our coast are still showing these signs. So it starts with this abnormal twisting of arms. Then there are lesions. Then the arms fall off. Autotomy, and severe tissue degradation. And then a lot of these stars obviously have been dying. And so these are just a few of our pictures of the dominant star. So this is the Ochre Star, which is a dominant star on certainly our shores in the San Juans, and I think used to be, here? Yeah, here. So they're not always ochre, sometimes they're purple. This is the twisting. Lesions beginning, and then just to give you a sense of how this sort of grim situation progresses. So this was the first record we had of this. It was from a diver called Neil McDaniel, and he posted these pictures of a big die-off event in British Columbia. And so he'd been photographing this rock outcrop on October 9th, 2013. 2 weeks later, this is what it looked like. All of these giant sunflower stars basically went through the process we've talked about, fell off, and basically these are just piles of the spicules from those stars left behind. So this was pretty dramatic to have an event so rapidly starting. Plus, these are stars that were kind of in the prime of life. They were fully reproductive. So it was a kind of a dramatic event that we were watching. I'd been studying some diseases as part of our research coordination network at Friday Harbor Labs, and so we were immediately interested in trying to isolate what might be causative for this. What we had no idea about at the time was that this was going to spread and affect so many species over such a wide range. We've seen epidemics in stars before. Usually it's a single species. And it's usually a very small geographic range. So most often in Southern California, and a few times in our waters. But we'd never seen anything with such a wide host range. So that's what makes this unusual. So what causes sea star wasting disease? When this first started, there was enormous arguments among scientists about what the cause was. There were those that were convinced it was a low oxygen event, a hypoxic event. There were others convinced that acidified waters were to blame. Still others were really sure it was radiation from Fukushima. So there were all sorts of ideas and passionate beliefs about what was going on. This is work that a colleague of mine from Cornell, Ian Hewson, led. And the amazing thing about this is a lot of the kinds of diseases or the causative agents we find in the ocean tend to be bacteria, sometimes they're fungi, they're parasites, not as much that we've been able to study viruses. And so in this case, we were very fortunate to have this virologist, Ian Hewson, at Cornell, who had been studying echinoderms, which is what sea stars are, and put in 4 months of incredibly hard work to isolate a virus that he thought was causative in this case. So we set up experiments in Washington State to actually demonstrate that this virus was the causative agent. And so I'm going to take you through the steps that Morgan Eisenlord and Colleen Burge went through to kind of demonstrate that this was the cause. So yeah, I mean, that looks sad. So what you have to do is start by grinding up a 6-star and creating a filtered version of the exudate that's viral sized so that you take everything else out. Then one of the treatments is the live virus size fraction. The other is a heat-killed version of that so that it can no longer be active. That's the control. Stars are injected, and we had to have special quarantine conditions to do this. This is not something we wanted to do anywhere where there was seawater running back into the ocean. We didn't want to be taking any chances, so we were established at a fisheries lab for this. And then it was a pretty exciting day. They actually went through several repeats of this with nothing happening. And then finally, one of these runs worked where the stars that were injected all got sick and the controls all did not. That's why they're smiling. And the next part of this is to actually do what's called a passage and to actually take these same stars and run it through an increased replication of stars. So in this case, we had 10 in each case. Same situation, same results. So this was a really key experiment that these two ran to actually be able to demonstrate that it was a virus that was involved for one of these species. Of course, there's a lot of other species. I really love these field guides that Neil McDaniels produces because it reminds us of the incredible biodiversity and how beautiful these stars are. These are just some of our pictures of some of the species that have been affected in the San Juans and further south. So one of the not surprising but interesting aspects of this is that there's such a wide range of hosts that were showing signs of infection and variation in susceptibility. The reason that we did the experiment, with the sunflower stars, that's the most susceptible. So that was the species that we were seeing, in 2013 and 14, very rapidly dying, whereas these other ones were showing signs but going down a little bit more slowly. So this was the logical one to work on first. So what causes the wasting disease? The best candidate pathogen is the sea star-associated densovirus. Densoviruses are also found in other echinoderms. And this is some of the interesting work that Ian Hewson did to sleuth this out, because of course we had lots of questions. Was this new? Where did it come from? And it turns out that this particular densovirus has been found in museums for over, museum specimens that are over 60 years old. So it's been in our waters. And that may surprise you, but it's actually not an unusual thing that a normal constituent of our marine ecosystem sort of ticks along, isn't causing a lot of trouble, and then something changes in either its genetics or in the environment, and then an outbreak starts. So we still don't have sort of the answer to that really big mystery. What is it that's changed? Why did this suddenly happen? We think that part of the mystery probably lies in the genetics of that virus, and that's work that Ian Hewson's group is trying to understand now. Parvoviruses in general infect many different organisms. You may know, those of you that have dogs may know that you always vaccinate your dog against parvoviruses. So that's a completely different, unrelated virus, but it's in that same group. This is a picture of the ochre star that I took in Bamfield Marine Station, sort of just as this outbreak was occurring, and it's just kind of a reminder to me, and I hope to all of you, of how beautiful this biodiversity was, sort of before a lot of these are disappearing now. Certainly, there's not these numbers in most of our sites. These were relatively healthy when we visited. Maybe about 5 or 10% of them were starting to show a few signs, like this one. But by early August, at some of these sites, up to 50% of them were showing signs of the disease. So I want to show you some of our data from the San Juan Islands, not too far north of here. Before I do that, I almost forgot. One of the things that we do in addition to, well, let me back up why we would do this. So this is an investigation of the immune system of the sea star. And a lot of people sort of ask me, well, what can you do? Okay, that's great. You figured out there's a virus that's causing this, so that's good. Now what do we do about it? And there's not actually a lot we can do once an agent like this is in the oceans. But probably one of the first steps that anybody who's concerned about sort of endangered organisms or risks from disease is to think about captive breeding programs. And in order to do that well, you have to know something about the immune system. So this is a study that we did with our students where we actually compared the gene expression of stars that were injected with the virus versus control stars that weren't. And I'm not going to take you through all those words. They're deliberately kind of very small and fogged, so you won't even try to read them. I just want you to see that all our treated ones, these three here, here's the ones treated with virus, here's the ones that are control. Look at all those red colors. So those all have to do with the genes that lit up in response to being injected with that virus. So that's the immune system of the sea star fighting back against this. So there's a lot that we can do still in terms of researching and understanding the natural mechanisms of these organisms in fighting off disease. And this is kind of the first step in that. So a little bit more about the surveys. We have a combination of surveys. We did a fairly active citizen science program because we just needed information from a very wide range of areas about the condition of the stars during this event. And so I'm going to show you a little bit of the data from the citizen science and then more of our own sort of researchers' data. These are a couple of our students doing surveys. Let's see if I can get this guy to start. So what this is the citizen science data, and it'll show you an animation of how the diseased animals showed up at the different sites in our region here in the San Juans and out to the Olympic National Park, which is actually one of the first places that this was recorded. So you'll see colors showing up. Green means that there's no disease, and obviously as it gets redder and redder, it's higher and higher prevalences or percentages of the population affected. So let's run this and see how this goes. So there's the first one, summer of 2013, pretty rapidly followed. You can see a lot of places looked pretty good, even into the spring, and then bam, summer 2014, that was basically the time of our really huge outbreak. Not as many, I'm going to play this again because that's really fast. So what you saw in this is that sort of a really sort of slow lead up to the outbreak in our areas. What I don't have is your data. Down here, there was a massive outbreak during the fall of 2013. So it came much later to our waters. And I'm going to come back to one of the things we think about why that is. So just to run this again. So there we're starting here. We looked, everybody was good, pretty much bam, everybody was not good. And then there's a lot of X's, which mean these are all sites where we've checked and there aren't any individuals. Last spring we were a little bit hopeful, all those sites were green, the few remaining stars weren't sick, and unfortunately this summer there was another round of it. So I'm going to show you a little bit of data from this paper that we have that's not published yet, it's in review. And it talks about sort of the impact in our waters. So this just gives you an idea of the proportion. This is back to that term prevalence, the percent of individuals in a population that are affected. And what you can see here is that over 50, 60, 70, 90% of the stars at all these sites in the San Juans were affected. There's a few places where they did a little bit better, and not so many were affected, and on the outer coast. We also followed these individuals through time, and unfortunately that was not really a happy thing to experience. So these were all of our 12 sites, and basically all of them went down really dramatically between sort of the end of June and the middle of August in 2013. So a very rapid epidemic. And then this is just one site to give you a sense. This is a site where we had several thousands of stars. This is the increase in that prevalence. Whoops. That's the increase in the prevalence to August. And this is the crash in population numbers from hundreds on our transects to just a few. And then we still have this other little pulse this year, so it's continuing. And this just shows, I won't go through the details of all these individual sites. So some of the things we're interested in is the role of the environment, and this has been one of the trickier things for us to figure out is temperature. So what's shown here is the probability from our surveys of a star developing the disease at different temperatures. So different sites had different temperatures. And what you can see here is that there's a very strong signal of these stars having a much higher probability of developing the disease sooner on a steeper trajectory at the warmer temperatures, this trace here. The other thing is that the other probability of developing disease that was very strong is the size. So again, this is showing the individuals that were in the sort of 200 size, so these are stars about this big, had a much higher probability of developing the disease early on. And the littler ones, a much lower probability. So then what we did is, this was just the field survey data. We wanted to confirm that temperature was important. Obviously, we were very interested in that, not only from a lot of my other work, which has been with diseases that are temperature sensitive on coral reefs, but also because we were under such warm conditions basically the last 2 years, and with this year being the absolute worst. And so what we did is we grew them at 4 different temperatures in the lab, both the juveniles and the adults. And what you can see here is that the adults, so this is percent healthy, so we have 100% at the beginning, and that changed very fast, with the adults dying much more rapidly than the juveniles. But in both cases, it was dependent somewhat on the temperature that they were grown at. So let's see. And then this shows the time before onset of disease and death plotted against temperature. And again, you can see that at lower temperatures for the adults, it was fewer days than at the higher ones. So a role for temperature. So that's sort of the story that we have to date on the Ochre Star. This is just one piece of a much broader range of investigations. So this is the work that we've been doing. As you know, there are scientists working all up and down the coasts, sort of contributing different parts to this story. I also want to expand the discussion a little bit to another species. So the Ochre Star we focused on intertidally because it's so abundant in our water, so we could get the best data. It occurred at a whole range of different sites. The Sunflower Star is a subtidal species, but remember that was among the most susceptible. So that was kind of why we picked the Sunflower Star for those injection experiments, because we expected it to respond the quickest. I'm afraid that the current estimates of their numbers in the natural environment are very poor right now. So they're completely absent from the San Juan Islands in our surveys in the spring, summer, and fall this year. In addition, CDOC Society did a survey of over 100 transects this fall and didn't find any individuals at all, where normally you'd probably find like 3,000. We have reports from Howe Sound, which is just north of Vancouver. They're almost completely absent. They found a couple of really small ones. Banfield, British Columbia, on the outer coast of Vancouver Island, they're absent. The reports I've heard from Monterey are that they're absent. Sitka, Alaska, from several sites where they were abundant, they're absent. And where we do know that they're still occurring and they're healthy are in the Aleutian Islands. They're healthy Pycnopodia as of this fall and last spring at Kodiak, Ian Hewson went. So this is a pretty big impact over a large part of the range. So when we start talking about biodiversity, most of these sea stars are what we call endemic species. So the only place on the planet they live is on our west coast, so between Mexico and Alaska. So it's good news that there still are healthy Pycnopodia, the sunflower star, in the Aleutians, but it's pretty concerning news that they're absent for a lot of different locations along their range. There are occasional small pockets. So what do we think the role of temperature is? I really can't answer. I shouldn't have worded it that way as if I was going to answer it. I can't actually answer that question, but I think it's really important to realize that we're under very unusual warm conditions in our oceans, or at least on our Pacific coast. This was a report from the New York Times last week. They entitled it "The Pacific Ocean Becomes a Cauldron." And they're showing this strong El Nino of warming water. This is plotted as the departure from average. So you can see this is up to plus 9 degrees Fahrenheit, which is pretty big departure. And then you can see our coastline here. The water is hot, and it's been really hot for quite a while in our coastline. So there's no indication that the warming temperatures that we had in 2013 actually triggered the outbreak, especially because that outbreak continued into fairly cold waters. My interpretation of the temperature effect is that it probably made the mortality more rapid and perhaps worse than it would have been. And it's the kind of outbreak we would expect under these kinds of warming conditions. This is just a little bit more, our actual data on the disappearance of the sunflower stars in the San Juans. So this is, again, that prevalence. That's the percentage of individuals that are sick. So when we did our first surveys in May of 2014, everybody looked pretty good for the sunflower star. In June, we started to see sick individuals reaching up to almost 50% of the large ones in July. Everybody kept hoping the little ones would stay healthy. What we had observed with the ochre stars is that we rarely surveyed sick small ones because in the lab they died so quickly. And so similarly, we think that may be partly the case with these guys. And then into August, virtually all of them were sick. And then the numbers in the next year are essentially a high number of them sick, and then they're virtually gone as of 2015 in our waters and in quite a few other places. So I want to make the case that sea star wasting disease isn't alone. I mean, this is an anomalous outbreak in our waters. This is a very large, as I said, this is the largest wildlife disease outbreak that we've recorded. But of course, we see other disease outbreaks and that affect biodiversity and that affect some of our marine species. So these are just a few examples. This is abalone. So the wasting disease of abalone has driven 2 species of the California abalones to endangerment. So again, that's a bacterial disease in this case. So, and there's also a temperature component to that one. We've been working for many years. Most of my work till this point has been with corals, and several species of Caribbean coral are now considered endangered, and infectious diseases have been partially to cause the cause of that. There's starting to be a big outbreak of what we call a shell disease in lobsters on the East Coast that's also quite temperature sensitive. and is starting to take a fairly big toll on those populations. And then finally, the work that we've been doing in the San Juans, in addition with the sea stars, is with it's called a wasting disease of eelgrass. It's not in any way related to the sea star affliction. This one is caused by a protozoan and not by a virus. But again, it's one of these that's temperature sensitive and that's sort of affecting our marine species. So one of my other hats that I wear, one of my jobs, is to be the lead investigator on this project called, it's a research coordination network that brings together about 50 scientists around largely our country, but also some European and Australian scientists, to actually try to understand in particular the environmental facilitators, as well as the causative agents of some of these infectious marine diseases under changing ocean conditions. So one of the ones that we've done a lot of work with are the corals, but certainly we now add the sea stars to this project and the eelgrasses. And what this does is it allows us to bring a large team of scientists together. For example, with the Sea Star event, we were able to move pretty quickly to help isolate and to provide Houston with the samples because we had a whole team of researchers that were working together. We were able to access lab facilities at a fish quarantine lab as part of this research coordination network. So we think this is important work to do because diseases in the ocean are kind of out of sight and out of mind. It's not something you can actually see happening. The Sea Star event is very unusual in that it happened on the public beaches and where people could see it with an iconic species that they really cared about. And so compared to most of the outbreaks that we have, it was quite unusual. I want to just sort of continue this theme about, although this was an unusual event, it's not beyond our experience. So probably many of you know that there currently is, and over the past 10 years, there's been a big outbreak of disease in frogs and other, it's largely frogs, but also some of the salamanders as well. There's been a lot of work to try to disentangle the same kind of terms, host, pathogen, and environmental determinants of this recently emerged wildlife disease. So this has been going on for about 15 years. And this just shows you sort of on our continents the distribution of frogs that have this particular disease. Now, they're not in the ocean, so I'm not an expert on these, but I do want to kind of draw your attention to sort of, what the outcome has been. This was kind of a newly discovered disease about 15 years ago, and I'm seeing papers like this. This was a recent paper, actually, that just came out last week, estimating that about 3% of frog species have disappeared. That means an entire species that's gone extinct. So on these, this was a really complicated statistical paper sort of estimating the grand total of extinctions to be about 200 species. This is a staggering impact from a disease outbreak. And so I think it's important to realize that these infectious diseases can in fact be very potent changers of our biota and things that we really do need to understand a little better, particularly in the ocean where we're a little bit behind in terms of having some of we don't have a CDC for sea stars currently. We don't have some of even the facilities that agricultural systems have. So there's a lot more. But we do have a lot of interest. We do have people that care about what's going on in the ocean. This is an example of a policy action that was activated by this particular outbreak. So we were contacted by Congressman Denny Heck from Washington State, who said, what could we do better? How do we handle these kinds of situations in the ocean? And working with his team, we helped him put together words around the the Marine Disease Emergency Act, which he then introduced, and it's still sort of kind of sitting there being worked on. And what this kind of legislation will do is provide funding for cases like this when there's a surprise outbreak in the ocean where there's a lot unknown. For example, I would estimate that all of the scientists that worked on the sea star wasting epidemic probably racked up, tens of $millions worth of research, and yet that funding wasn't there. And so this was a lot of work that was done by kind of bootlegging and patching together other grants, folks working far more hours than they should have just because they really were cared about the work. And so this is just an example of something positive that can be done. So just to summarize some of the things I've told you, the smaller ochre stars are slower to infect, and these were from lab experiments, but they die really rapidly once they become sick, and so it was very difficult to detect sick ones in nature. And this, some of you may have heard some really hopeful reports about, oh, there's a bunch of recruits, we think they're going to be okay, none of them look sick. Unfortunately, a lot of those did end up dying and not sort of getting to the next level, and we think this is probably why, that explains that. The ochre star populations I've shown you have been heavily impacted. We found over quite a few of our sites, over 90% of them disappeared between 2013 and 2014, and that's continued into this year. So we don't know how long it's going to take for the recovery. We do feel good that there's still ochre stars on our shores, and I know there's still some here, so that's a really great thing. It's not as big an impact as with the sunflower stars. The sunflower stars are among the most susceptible, and they're certainly extirpated from some parts of their range, and we really don't know what the outcome is going to be in terms of recovery in this species. So I guess there's a few issues to consider. One is that disease can drive species to endangerment, and we have good examples of that, unfortunately, with the frogs actually being driven to extinction, and with abalone corals where species are actually pushed to the edge of endangerment. We do know that warming temperatures affects the rate and impacts of mortality. As I said, we don't attribute this outbreak to the record-breaking warm waters, but it is the kind of outbreak that we could see more of in warmer waters. One of the big questions is how many or which of these survivors may be resistant. We sort of have suspicions that some of the ochre stars could be resistant since they're still out there, they're surviving. And that's a really hopeful thought, that some of these are resistant and are going to be sort of breeding more resistant individuals. So, sometimes when I give this talk, it gets a little grim. And it is grim. I mean, I think there's so many people here because you're concerned and you're worried about not only losing iconic species, either temporarily or longer term, but also, just what else is going on in the ocean that you should be concerned about. I think we need to remember that our oceans, especially in this country, are actually healthy and vibrant. And these healthy and vibrant oceans really do need you to be kind of connected, either, through contributing to monitoring efforts. You're here tonight, you're involved with this incredibly amazing marine lab that's here right in your community, that's sort of involved in research, in the Bay. And I think that's kind of one of the most important things, so you should all be congratulating yourselves for, your level of support for this effort here. Another thing is support legislation that provides resources for monitoring and responding to marine diseases. And, it's not just sort of sea star outbreaks. We're having problems with El Nino causing shortages of food. For example, some of the birds, I know there was an article, this week about problems, mortality events with the birds. So there's sort of a lot of issues that require you to be informed citizens. And then I want to just remind you when I say, the oceans are healthy and vibrant, you're going, oh, come on, that's not what you just told us. You've been showing us dead things, and they're dying, and they've got diseases, and they're not going to come back. And so I just kind of want to remind you that actually we have a lot of bounty in our oceans. And so I want to tell you about one of my other projects that's a little bit more hopeful. And one of the reasons why I do that project is because I really do feel we have amazing biodiversity to celebrate in our oceans, despite the fact that I do this work. So this is this book that you were hearing about earlier. It's called A Sea of Glass, that's coming out this spring. And it's a search for the Blaschka's fragile legacy in an ocean at risk. I think it's fair to say we do have an ocean at risk. And the theme is that marine biodiversity is as fragile as glass. Well, what does that mean? Has anybody ever seen the Blaschka glass flowers at Harvard? Okay, okay, we've got people here. Okay, so this is a very historic glass collection that was made 160, well actually the flowers were about 140 years ago for a collection that's very much prized at Harvard. Well, Cornell has the Blaschka glass invertebrate collection. And just to get a sense of how exquisite these are, it's a little bit hard in this picture to know which is the living swimming anemone that lives in our waters in the Pacific Northwest, and which is the glass one. So this is the Blaschka glass. So the father and son team crafted over 560 different species that we currently house at Cornell of these glass pieces. And so that's why I call it a sea of glass, because these are all glass. They're all exact replicas of particular species that are in the ocean. And so that's great. You're thinking, well, what's so hopeful about that? They're all glass. What can we do about that? This is even worse. Well, the good part of it is that what we've been doing for the last few years is going back and sort of using this as a time capsule and seeing, can we still find these? Are they still in today's oceans, right? They were made 160 years ago. And, the really exciting thing is that a lot of them are still in our oceans. Sometimes they're a little hard to find, but a lot of these, they picked fairly common species that are still out there today. This thing is just a troublemaker. So each of these things in glass is made life-size. It's not like as big as that, but the model itself is actually about that big. And but in nature, this thing has tentacles that stretch probably 30 feet behind it. So and it can form these big swarms. So that's not always a good thing, but it's really beautiful in glass. So that's what some of those look like. And of course, we do have an asteroid, and this is a beautiful picture of the glass by Guido Motafico, who's a very famous photographer, and courtesy of the Dublin Museum, who hired him to do some of those pictures. So when I say that, it's really upsetting. This has been a very challenging time with the sea star epidemic. It's something everybody needs to pay attention to, and well, maybe not everybody, but I hope a lot of people, I'm really pleased to see people are interested in it. We need to think about the changes to our ocean and what we can do to safeguard and maybe do things a little bit better. So I just want to finish by thanking other collaborators on this project, many of whom have their own research programs, so I've been telling you about some of the work we've done. There are other scientists, for example, Santa Cruz has, Pete Ramundi's group has really led the way through the monitoring programs along the entire Pacific coast, and that's been key in our sort of studying this whole outbreak as it's unfolded. Ian Hewson has been teaming up recently with the Seattle Aquarium to do more work on the genetics and other aspects of the continuing outbreak, and as well the Vancouver Aquarium has been an important collaborator in providing us information about where different outbreaks are. Those are just some of the main people. There's been a huge group involved. This is, again, a large outbreak, our entire coast, and so there have been a lot of collaborators on this. So with that, I think I'll take any questions.
So I was just reminding Drew that one of the neat things that we do here is that we have our graduate students interview our speaker for a few minutes, and then we open up the floor for public questions and answers. So we're going to just take a short break, not a break, a short minute to raise the screen and have our student interviewers come up. I'm also going to introduce you to our associate director here at the Romberg Tiburon Center, Dr. Ellen Hines. She's a professor in the geography department, and she's also the coordinator of our graduate program. And she's going to introduce two of our graduate students. And Drew, there's a chair for you as well. So we're going to warm you up and hope that you keep track of the questions you'd like to ask, and then we'll take them in a few minutes.
Hi, everybody, and welcome. It's so glad to see you all here. So we've got some grad students here that are going to just grill Drew. It's going to be brutal, but I think you'll all enjoy it. So first on my left here, we've got Ryan Hartnett. Ryan Hartnett's in Karina Nielsen's lab. And both of these students are products, even though students aren't really products, of California State University schools. So Ryan here got his BS in general biology at Sonoma State and is currently completing his MS in marine science, interested in large-scale climate, oceanography, ocean chemistry, and how that relates to marine ecological interactions. He's interested in working in marine technology development or in oceanography research. The title of Ryan's thesis is Connecting the Dots in the Gulf of the Farallones, Linking Physical Ocean Conditions to the Ecological Success of Planktivorous Predators. Sexy, huh? All right. This is Kara Gallagher over here. And Kara works with myself and John Stern and Sarah Cohen. And she got her CSU degree at CSU East Bay in biology, first college graduate and her student, and the winner of our College of Science and Engineering Women in Science and Engineering Award last semester. So we're all pretty proud of Kara over here. Kara got her MSc, is getting her MSc. She's going to graduate this semester. Sorry, in ecology, evolution, and conservation biology, interested in ecological modeling as well as foraging ecology of marine predators. She's going on to a PhD, and she's going to pursue a career in teaching and research. The title of her thesis is Modeling Energy Sequestration and Foraging of Harbor Porpoises in San Francisco Bay. Okay, without further ado, Ryan, you have some questions for us.
Yes, I do. First, Drew, I just want to say thank you for that wonderful talk. It was very informative and interesting and impactful. Your work on this epidemic is in depth and encompassing, and it was a privilege to be here to hear her speak tonight. Absolutely. This particular issue is near and dear to me because when this outbreak began, I was in my marine invertebrate zoology course at Sonoma State and out in the field, and it was very disheartening to see this tragedy unfold in front of my eyes. So now that you have identified the virus that is the culprit for sea star wasting disease, what are the next research steps to address this epidemic, and is it possible to develop a vaccination for susceptible sea stars?
Ooh. Yeah. How about that? Oh, there we go. Oh, that's a good question. I think at this point, I'm speechless. The idea of developing a vaccine Probably not, although, we always have to keep in mind that there's definitely amazing wizardry that goes on in terms of medical technology, and we do have a vaccine for parvovirus for dogs, so I don't know. Maybe there's some potential for it, and that could certainly be helpful with captive populations. I think probably the more immediate sort of response to this is to look for resistant individuals. And what I've heard some of the aquariums talk about is the possibility of breeding for resistant captive populations. And so that's certainly been the strategy with amphibians, for example, with the frogs. There's projects called ARCs that sort of like Noah's Ark that are trying to breed and sort of keep safe resistant individuals. So that's kind of what I would think, yeah.
Cool. Excellent.
I also want to thank you so much for that fascinating talk. I have a bit of a cold, so you guys can probably hear that in my voice. As somebody who's also been able to spend some time at Friday Harbor Labs, I was there between the summers of 2010 and 2012. I can remember going up there and diving and seeing the sea star Pycnopodia absolutely everywhere. I actually had a pet Pycnopodia that I used in my research. His name is Pat, or her name is Pat. We don't know. And now hearing that Pycnopodia are so drastically impacted by this disease, it's really concerning. So I was wondering, what makes one species more susceptible to a pathogen than another species, a closely related species? And also, why are sea stars being hit by this so much more drastically than other species of echinoderms like sea urchins and brittle stars?
Oh, those are great questions. Boy, grill me, they are, yeah. I think with the Pycnopodia or the sunflower star, I mean, these are the ones that are about this big across. I mean, they really are spectacular, and you probably saw like 5 or 10 on a dive when you were there, because they used to be very abundant. We don't really know why they're that much more susceptible. One possibility is that they have, it may be the connective tissue. This particular virus seems to attack the upper surface, or what we call the epidermis of the stars, and the Pycnopodia are really much more soft-bodied. So one possibility is some of these soft-bodied stars are tending to be a little bit more susceptible. So it may just be that it's easier for the virus to get in, or they don't hold up quite as well. You also asked that other really good question about, is it just the stars that are affected, and do we know how wide the host range actually is? To this point, we think it's mostly the stars, although you probably know that there have been some outbreaks among some of the urchins as well. So urchins are close, kind of coming in and out.
Should we trade? Yeah.
Let's trade. So, yeah, that's better. Urchins are close relatives of stars, and so we've been kind of watching those, and there have been outbreaks of other diseases in urchins along this coast anyway. I don't know if anybody's confirmed whether it's densovirus or not. The other group to look in are the sea cucumbers, and mostly they seem to have been holding up okay. So as far as we know, it's just that class called the asteroids. Yeah. Did those all your questions? Yeah. Okay, good.
So Drew, I know you're a coral reef pathologist as well. So I was wondering, in the tropics, there's a sea star called the crown of thorns, another large sea star, and they're responsible for the decline in coral reefs in many areas. And crown of thorns abundance varies naturally, and when high, they can eat and kill a bunch of the reefs, or the corals that build the reef. It is thought their abundance is increasing more recently due to human-caused nutrient runoff. And I was wondering, are the crown of thorns sea stars sensitive to sea star-associated densovirus? And if yes, would it be ethically responsible to use the virus to control their population growth? And has this possibility been discussed among the scientific community?
Well, that's an interesting question, Ryan. What do you think the answer is? No, we're not going to introduce densovirus to control crown of thorns. But we may get a chance to observe the natural experiment because there is a new outbreak of disease in Australia that started in Melbourne. It's not confirmed yet whether it's densovirus or not, but it looks a lot like it. And so it would be spreading north now. So far, although this virus has spread to other shores, it doesn't seem to be hitting the tropical species as far as I know. And so that raises a very interesting question about whether it will get to the range of where the crown of thorns star is. However, if it gets there and if it kills the crown of thorns, everybody will be really happy. Because it's probably one of the major causes of mortality of corals on the Great Barrier Reef and other tropical waters. Yeah, so it's a good question, and there's a lot of interest in that.
So to continue on what you were just talking about, we do know that the sea star wasting disease has happened in other places and at other times, but this West Coast event that's occurring right now seems to be very extreme in its range and the amount of species affected. I was wondering if you had any ideas why the West Coast is such a target right now. And also, if related to temperature, do you think that increasing temperature could be increasing the virus's ability to infect a host or actually reducing the host's fitness?
So those are great questions. In terms of the temperature effect, I don't think we know whether it's affecting the host's immunity or the happiness of the virus. It is the case, though, that a lot of factors that compromise the host's immunity seem to make them get sicker faster. And so I'm a little bit suspicious that part of that temperature effect is sort of some stress to the host. For example, in places where there are more pollutants or there's more really bad freshwater runoff, we found sort of foci of the stars dying faster, sooner in those kinds of places. So it's a little bit similar to the temperature effect. The thing about the West Coast, is an interesting one because it's not just that this is happening on our coast. Okay, there's also this outbreak is occurring in stars on the East Coast, and in fact, it started on the East Coast in the Northeast with the Asterias species. The difference is that it hasn't been very well studied on the East Coast, and so we don't fully comprehend the magnitude of it. But I know that even just this fall, I was getting emails from folks in Massachusetts saying, "Oh no, another round! A bunch of our stars are dying again this year." So I don't know that there's necessarily a difference. What's spectacular and sadly striking on our coast is that we have such a rich biodiversity of sea stars. In the area of the San Juan Islands, there's over 85 species of sea star in our key. So that's an incredible biodiversity. And I don't know what the numbers are here, but it's probably not too much less than that. You don't see those all the time. A lot of those are deep sea or very rare. We don't know that it's only 20 species that are affected. Those are just the ones, that we see and can confirm. Yeah.
Thank you. You did a great job. Here at RTC, we're incredibly proud of our students. I know that I feel like when I can only do so much, but when I have students, it's like I have 5 more fingers and 5 more pairs of hands. So thank you very much. Karina?
You get to stay. Okay, you get to stay. You have to stay. We don't get to let her off the hook that easily. So this is where we open up the floor for questions from the audience, and we're going to have a couple of students who will bring a microphone over to you, and as soon as you have your hand up, they'll come over, and you can ask. Yeah, she really, really wants, yeah, we got an eager beaver, so here we go.
So how long have the sea stars been around for?
How long, you mean in terms of evolutionary time? I guess over 500 million years. Yeah. Pretty long time. I'm sorry, they haven't what?
They haven't like disappeared and came back. This hasn't happened before.
How many extinction events have there been? Well, that's a good point. I mean, a lot of species, we do certainly have, species going extinct, disappearing, and coming back, maybe not the same ones. Usually we don't see this many, affected sort of this quickly. Although you're raising a really good point, which is that we have almost no data or baselines about the impacts of marine disease over time. And so it's really hard to say what's a typical event, what's an atypical event. And so, those are...
No, I was just thinking if the water heated up and it happened and some survived and then they get colder again and they come back.
We hope they will.
Of course they will.
Yeah. Yeah.
Question, so the food chain, the sea star, other species affected by the virus in some way? What's the food chain surrounding the sea star? Who eats it?
Right.
What does it eat?
So, I mean, that's a really interesting question. One of the things that we were kind of speculating about early on is whether this virus, for example, could be concentrated by some of the shellfish that sea stars eat, so that it could actually be, concentrated in the food chain and, delivering them a bigger dose. So that's one aspect of that. I think the other part of it, though, is what will happen to our marine biota now that these stars are gone. Given that we think they're keystone species, are we able to actually pick up, a big impact? The reports that I've heard, I mean, our monitoring, our work has been to really focus on the stars themselves and the host pathogen interaction, but other ecologists that have been monitoring nearshore populations have been reporting big outbreaks, large numbers of urchins, so in our waters, which is not surprising that especially the sunflower star eats everything. So it's an incredibly voracious predator on all sorts of stuff, and so we're not surprised to see the urchins. The keystone effect of that ochre star is cited as mussels, and so the expectation is that there will be large recruitment and larger numbers of mussels. And I haven't seen the data on that, so I can't really tell you about that. The urchin impact is kind of the one that's talked about the most, because of course that can have a cascading effect in the food chain, because urchins are themselves voracious herbivores, and so when there's lots and lots of urchins, they eat all of the algae, which is not a good thing, because we like kelp beds, and kelp beds are important habitat for fisheries. So there could be some potentially bad impacts in terms of the cascading effects on our ocean food webs, but we don't have all that confirmed. And then just to make it a little bit more complicated, with these warming waters, there have been disease outbreaks reported in the urchins, sort of in the Southern California and maybe up to these waters. I'm not so sure about that. So, yeah.
Thank you for your talk. We've done about 2,000 kilometers of ROV surveys up and down the coast, and we've seen them basically lag far behind the shallow disappearance in the deep water, and then usually by about a year, and then they would disappear there too. So most recently, we returned from the area from Mendocino to the Oregon border, last year at this time, the Pysasters and Stylasters were plentiful, several thousand in our surveys. And this year we saw one. But from your graphs, it looked like that area was skipped over and things started disappearing in Vancouver. And I was curious why you think that preceded our loss of sea stars in the northern part of California.
Can I just, so for your ROV surveys, did you also do sunflower stars? And are they pretty much gone? Gone. From where to where?
The Mexican border to Oregon. In the deep water.
Wow. Which is where they are.
Yeah, from 20, we pick up at a diver depth. So we start at about 20 meters and we'll go down as deep as 600 meters. I mean, they're not living that deep, but so we're covering the full range of their normal habitat.
Yeah, so that's, I mean, it makes sense that it took longer. Yeah. So that's the sunflower star, and then some of the solasters too, probably, you're not finding very many of. Yeah. Well, I really look forward to talking to you more about that later, to hear more about it. There was a gap. I think what you were asking me is about why it took so long for the San Juans and Oregon. So it was a little bit strange and actually very mystifying to us in 2013 when a bunch of stars died in Vancouver, north of Vancouver, and then everything kind of fell apart from Monterey south in the fall of 2013. And then our stars stayed perfectly fine, we thought, until that following summer. So it took almost a year until, and we don't really know. One possibility could be water temperature. We had much cooler waters during that period. So the big mortalities in Vancouver were all the sunflower star. So that was kind of what had everybody's attention, and not so much the ochre star. So I'm thinking that some of that gap is explained by a combination of different species and then maybe water temperature. Now in Oregon, I would love to know what some of actually, your former advisor, Bruce Mengy, thinks, because there was a big lag there, and I'm wondering whether it's some of those offshore currents as well as water temperature, but we don't know. I think I wish we knew. Yeah.
I know it's brought up about Fukushima, but has anybody really looked at the way the currents go, and is there any relation to the currents of the Fukushima radiation coming in and maybe affecting these areas in different ways?
At the time of the outbreak, there was no evidence for radiation in our waters. This was several years ago, so that at the rate at which that was drifting. And there were a lot of programs, especially from the University of Washington, to be carefully testing the waters and the fish. And so there was no signature of any radiation.
About the same time. I mean, it took,
Yeah.
I think, 2 or 3 years to get to the West Coast,
Right,
which would be
yeah.
about the same time. that
Well, about, from the models of when there was going to be any radiation hitting our coast, that was long after the outbreak started. So the timing doesn't quite work out for that. The only thing you can't rule out is that, there was some kind of a 6-star that originated in Japan and came over, with the wreckage or something, that's a possible event that could have happened, but there was just no evidence for it. And again, the distribution of stars didn't really match that. So there was really, everyone pretty much ruled that out. Yeah. Yeah.
Hi, you said that you found the virus also in a museum sample of a Echinoderm from 1942. Did you compare the two? How has the virus changed?
I would love to know the answer to that. I'm not a virologist, so the virology work is done by Ian Houston's lab at Cornell. And it was his brilliant idea, really, to go back and look at those museum specimens. And they did find a virus that closely matched the denzovirus, so it sort of matched out to the same species. And it's a burning question in my mind as well to know whether there's been a difference in the genetic code from then until the virus that we have now, and I don't know the answer to that. But I do know that that's one of the priority areas that his group is investigating now. Yeah, yeah. And in fact, you know, to match up the phylogeny of that virus in all these species of stars, but we don't have the answer. But it's a good question. Yeah.
Okay, we're going to go all the way to your right in the back.
Hi. Great talk.
Oh, thank you.
So I'm interested in temperature and its play on stress and stress response. And so I'm wondering, is anybody looking at the transcriptome of a healthy sea star in different temperature treatments and seeing how it compares to the environment, or to our sea stars in the environment, their transcriptome, and then maybe understanding if they're stressed and if the temperature is causing stress, so maybe we can say for certain if it's a player?
It's a great idea. Or I don't know anybody that's doing that. I think it would be a really good thing to do. Yeah, there you go. There's somebody's PhD project right there. I think that would be a good one to do. Yeah. You know, there's been a lot of that kind of work done with corals, including in our group, following them through some, you know, really stressful warm events. And those studies are very valuable in showing you that, yeah.
Do you know the mechanism of transmission?
No, we don't. So one of the black boxes in general for disease in the ocean is often transmission. There's really very little known in a lot of these cases, and it's not confirmed yet in this case whether direct contact is required. I would just offer the observation that there were outbreaks in all of the major aquaria, Vancouver, Seattle, and Monterey Bay Aquarium, within several weeks of each other inside the aquaria that suggested the possibility it could be waterborne. So that's all we really know about that, yeah.
Hi, okay. So I'm wondering if the disease is communicable to sea mammals, specifically pinnipeds and sea otters. And if so, what would the disease look like in sea mammals?
Well, that's interesting. Well, so the thing about viruses, they tend to be pretty host-specific, which is one of the slightly strange things about this particular one, the fact that so many, you know, we've got over 20 species that are already affected, but they are all sea stars. So it would be a very, very strange aspect of biology if pinnipeds were infected. So that seems, the virologists are telling me, is a very, very unlikely thing. However, if it was, I suppose you might expect some kind of disruption on the surface. So one of the characteristics of this has been that it attacks the upper surface, what we call the epidermis, the outer skin of the star first. And so I suppose if I were going to look for an effect, that's what I would look for. But it's very unlikely, yeah.
I was just going to ask that, are there any models or sort of pathways that see for the rebound of populations after events like this, either in sea stars themselves or related species?
The most likely thing that happens in an outbreak like this is that there will be recovery, that the host will become resistant, and that it will eventually rebound. So that's certainly what we're all hoping for in this case. The fact that we're still seeing quite a few of the ochre star, even though their numbers have been, you know, really knocked back, I think is hopeful that some of those must be resistant, right? That they're still out there, and so therefore they should be able to repopulate. It could take a very long time given how wide the host range of this event is, that stars all the way up into Alaska are affected. It's taken a lot longer in Alaska, and I don't know if it's because of the colder water, but sort of, the susceptible species are, starting to show more and more impacts, like the sunflower star. The sunflower star, though, I just don't even know what to say about recovery. I think it's too soon to know what we can expect with that one, and I think I have a lot of concern about the levels that we're seeing on that one. Yeah, did that answer your question? Yeah.
Okay, so thank you all so much for your attention and staying late with us tonight. I know it's a weeknight, and I want to be respectful of your time. Let's give a round of applause for Drew Harvell and her wonderful work. Just wanted to let you know, if you want to have a cup of coffee or something, we do have a little bit of refreshments still before you head out. If you want to chat with anybody for a few minutes, we'll be here for a little while. But thank you all very much for coming, and we hope to see you again at our Spring Public Forum in April, and at our Discovery Day Open House also in April. Thank you.